Hello, everyone, and welcome to today's webinar, Turn Research into Knowledge with our comprehensive product family. We're about to get started here. Just want to give people another second to join. So if you want to go ahead and grab yourself something to drink, a coffee, if it's early in the morning for you, or for the European folks, afternoon coffee, feel free to go ahead. Robert, Chris, do you have a coffee in your hand or?
I do. Still early for me. It's just 7 a.m.
Yeah, I wouldn't want to start my morning without.
Yeah, I agree. I can't operate without it.
Okay, so I see people are joining in. So if you've just joined, welcome to today's webinar. We're really happy to have you here today and show you how our research product ecosystem can revolutionize your research process. We will be introducing 4 of the most important tools in our powerful ecosystem: Curedatis, Article Galaxy References, and our Word add-on. You'll find out how easy it is to discover, acquire, manage, and create scientific content with our Research Solutions product. My name is Julia Heason. Oh, no, the slides don't want to move on. Okay, that's not working. I'll have them back on in a second here, or Chris, if you want to pull them up while I introduce ourselves,
Sure, yeah,
that would be awesome.
I'll do it.
My name is Julia Heason. I'm a marketing manager here at Research Solutions, and I'll be the moderator of today's webinar. And today's presenters are Robert Ratloff, Director of Product Development at Research Solutions and founder of Curedatis, as well as Chris Atwood, pulling up slides right now, VP of Product Development and Implementations at Research Solutions. Welcome to the webinar, Chris and Robert.
Thank you.
And there we go. Thank you so much. Before we dive into the topic, let me just cover some quick housekeeping items. And also, I want to give a brief overview of Research Solutions in case you aren't familiar with the company behind the product that I mentioned yet. So about the housekeeping items, you're all in listen-in mode, so you don't have to worry about background noise or your background as such. If you have a question, we have live Q&A enabled. So on the Zoom bar below, you'll just click on Q&A, and you can, as the conversation goes, you can add all of your questions. We do have time at the end of the webinar specifically to answer all of your questions. So depending on the question, we might, just like add it in as it comes up, or if it makes sense to wait until the end, but be assured that we will answer all of your questions. And if you want to rewatch a specific part or share the webinar with your colleagues afterwards, we'll be sending you the recording right to your inbox a couple of hours after this webinar is over. So about Research Solutions. Research Solutions was founded in 2006, and we provide solutions that simplify, streamline how organizations discover, acquire, and manage scientific articles and data. More than 70% of the world's top pharmaceutical companies, emerging life science firms, and other R&D-driven companies rely on us for one-click access to scientific literature delivered within seconds right to the user's inbox. Now, with that introduction being said, I'll hand it over to Robert.
Thank you, Julia. Hello, everyone. So our topic today is turn research into knowledge, and today we are in the luxury position that we have a lot of information out there and a lot of data. So you see that in those example searches from Medline PubMed. So for cancer, we have this exponential growth, and almost 5 million results were returned from Medline, and heart diseases, over 1 million results. And also, the famous topic right now, artificial intelligence is also in the medical area really prominent. So we have this exponential growth of scientific information. So what people need, so knowledge professionals and knowledge managers, are smart tools to handle those information out there, to share it, to really make it a knowledge ecosystem with their colleagues and also peers. So that is what we try to solve today, or show you at least our ecosystem. And so hopefully we can give you some tips and show you our ecosystem. But maybe Julia, maybe for our artificial intelligence topic, we have a poll, I think. So could you share that with us?
Let me pull it up right now. So as Robert said, we have prepared a poll. So you should be able to see the little pop-up here right now. And we would love to know, what are your thoughts on using AI to scale your research process? the options here, as you can see, I love it, or I would need to learn more, or something completely different. Feel free, if it's other, to add it in the chat. We would love to know what you think about it. Okay, interesting. I see votes coming in. I don't want to spoil the answers yet, though. Okay. Let me end this. I feel like we have everyone in here. Thanks for participating, and let me share the results. Okay, interesting. So 44% of all of you said, I love it. 33 say I would need to learn more. And then we have 22 saying other. I can't see anything in the chat yet. If you want to add anything to, yeah, what your thoughts are, feel free to do that. Robert, what do you search process?
Yeah, so that's really interesting. So I was thinking also I could not vote in this poll, so that's why I would like to vote I love it, but I need also to learn more. Yeah, so that is I think also maybe an answer, because I think there are a lot of opportunities right out there for knowledge, for your knowledge research process, but also in a lab, also in the regulatory industry. And I think that that's good that people are like to use AI, but I think there is a lot of opportunities, but also risk. So I think that is what in the end will make the difference in these tools which we apply. And so you need to have a basic understanding of AI. I think that is very important for the future. Well, thanks. Yeah.
Thanks, everyone.
Next slide, please. So again, our webinar today is about the research workflow, mainly of literature. So here we can see our ecosystem, which is broken into parts. So we have a search and discovery step in the first place, so where we would like to find the information which is out there. Then the next part can be done in Curedatis, so the screening of relevant information. So we want to exclude or include relevant information. After that, we have our Article Galaxy tool, which will be introduced by Chris later in the webinar, which is therefore the retrieval of full text documentation. And then after that, if you have all the information retrieved, so you would like to manage those information, you can do that in Curedatis, which you will see in a minute, and also in Article Galaxy. And then if you want to write up your research findings, you can do that in Article Galaxy and Curedatis via our Write and Cite add-on. And in the end, you would like to know or monitor your searches. So that is what you can do in the Article Galaxy alerts, or in the Curedatis alerts, and also in the Article Galaxy RSS feeds, so that you are on top of your research. So that is what we are seeing in the monitoring. So maybe for post-market surveillance or competitive intelligence, if you want to monitor products from your competition or your own products in the literature. All right. So that is my turn to hand over the screen.
I'll stop share here. I turned orange again, so there we go.
Screen will come up in a second. All right. So what we can see here is Curedatis, our new literature review system by Research Solutions. So what we have here is my library of projects. So we are starting from a setup phase. The next phase is then search. So that is very similar what I've showed before in those in the research flow. So the search, then we have the abstract screening, and then retrieve the full text of relevant abstracts. And in the end, we have a smart summary, which will provide you some insights of your screening. So we start here with a project, which I have already started to demo it to you. So basically, what we can do is do a search via PubMed. Maybe that's TKA, so total knee arthroplasty or a total knee replacement, and maybe rehab, rehabilitation. Oh, hard to write. And then we do a search. And then it returns a lot of results, and maybe want to only focus on randomized controlled trials in this area. And maybe for the last year. So very recent ones. We can now select all and check the import. So right now we would like to incorporate all those information into Curedatis. And you see, I've already done some searches. That's why we found some duplicates here. So we have an automatic deduplication in this step. So we can confirm the import, Curedatis. And then what we see here is we have an import history. So you can monitor all the steps you have done in your project, and to have a transparent overview on audit trail, so that you can go back and see what you already have done and how many records have been identified and how many of them were duplicates or valid. Yeah, so that is what we can do here. And what you can also do is you can create an alert here for your maybe post-market surveillance process. And then those results will be piled up here in Curedatis, so you can view the new results and check them and import them again. So that will be an ongoing cycle. So it will be reproducible as well. So that is for most of our customers in the regulated industry, like medical devices or pharmaceutical companies, a nice workflow to keep up to date with their findings. All right, and beside PubMed, we can also import RIS files from diverse set of databases like Google Scholar, Embase, and also from other reference management tools if you would like to import your citations here. So, and then if we move to the next part in the abstract screening, you will see your imported results here. So we can start the screening. And for the first time, you see our AI capabilities. So our AI model here recommends the PICO terms, so the population intervention comparison. So for instance here, patients with a mean age of 68 were included in this randomized controlled trial, and those patients had osteoarthritis and arthritic diseases in the knee. So that is picked up by the AI. So underlined means recommended. So you can, if you click on that as a user, then it will be confirmed by a human. And also maybe the intervention was the computer-assisted surgery, so the CAS, and the conventional surgery. So this is all done by AI, and this is also finding some outcomes of the studies. So here you can see that a user can really focus on the main terms in the abstract, so to find if this is useful for the review or for the research, and then can add this to the data extraction, which we see later. So all confirmed annotations, so you can all confirm at once here, confirm all annotations. So they will go in the data extraction part later, what you can see in the summary. All right, so then we want to include this one. And the next one is about a hospital inpatient versus a home-based rehab. So this is also recommended here, and we can confirm this as well. But you can also add your own labels here, like a study design label. So I put a dictionary of study designs here, so randomized controlled trials. So that is, the user can also, beside this PICO terms, can also add specific terms, maybe product names, any risk or benefit for their devices, or pharmaceutical drugs, and also could annotate something like maybe the study ID here. So, and this will also be extracted later in the report. And you can also add text to your document, like maybe it's a state of the art. Is it talking about the performance data of the trial, safety, or if you're working in a product management, marketing, you can maybe grade your articles, like was it perceived as positive, neutral, or negative. So that is what you can also do, or you can also extract marketing claims out of those abstracts or later in the full text. All right. And for your review, you can also provide exclusion criteria. Maybe like to exclude this one. And here we go. All right, so that is our abstract screening part of Curedatis with these AI capabilities. And if we move on to the next stage, so the full text screening. So I have already included some abstracts here. But if we want to go to ordering a PDF, because that is the next step, we have an interface with our Article Galaxy tool. So those publications can be sourced inside Curedatis, so there's no need to jump to the publisher site pages and buy them via credit card. So you can really use Article Galaxy and stay in one system, and this will then order your publication. So we will place the order right now. And most of the publications will be delivered in seconds. So we'll see that later. Maybe we start with one. It's not here, so that's what I did not include. And you will see the native PDF with the research findings. So here you have a PDF annotation tool, so you can mark everything what you want. Maybe we want to highlight the full study design here and add a comment here. So that is about study design. And this will then also extract it later in our summary report. So that's what you can do here, or provide comments for your colleagues or for your own risk. So, and yeah, so that is what we provide here at this step for the annotation of your PDFs. And you can, again, add tags or comments to the full text. So if we include this publication, so then our system will ask, do you want to praise the publication? So do you want to grade that? Maybe we have here an appraisal question catalog of, is the article suitable for your research? So the user can then decide which questionnaire they want to add here, and then they can grade the article if this is necessary, and they can specify what they would like to answer here. So we can confirm that, and this will also go into the summary later, you would see in a second. All right, and if we then move on to the last part in Curedatis, so now we are going from abstract screening over full text screening. So you extract your data and decide which articles and information is relevant for you. So then you see that later in the summary, we have maybe we can add the annotations here, which we did, or tags, any exclusion criteria. So that is then we are going from unstructured text to a structured output, what we can see here in a table-like format, and you can export that in a CSV file and then prepare your research report easily based on the PICO terms or any labels you did define during the screening process. So, and maybe also a summary of findings or the study ID. So that is what you can have here, and also the citation, yeah, so like the authors and journals. And additionally, so we have also the appraisal table here. So that is also what you can get out of Curedatis and store that in your research report or clinical evaluation if we are talking about medical devices and other dossiers. And last but not least, we have also the Prisma flowchart. So here you will see an overview of your screening and identification process. All right, so that is what I can show you today for Curedatis, but if there are any questions, so I think, Julia, we have time later for answering those questions. And yeah, Chris, I would like to hand over to you to show us our capabilities for Article Galaxy and our other tools.
Yeah, perfect. Thanks, Robert. I'll share my screen when you are able to stop sharing. I'm going to bring this piece up here. And,
as
ahead,
Robert mentioned, if you have any questions that come up, feel free to go ahead and add them in the Q&A feature, and we have dedicated time in the end to answer them all.
Yeah, definitely. I'll do my best to not keep this too long-winded for sure. Really what I want to talk about is the subject really, or the topic here is, turning research into knowledge and how our tools can help you with that. So what Robert really just spent some time discussing is how Curedatis can simplify and streamline the literature review process for you all, for organizations, for groups within organizations. And really what I'm going to showcase is how Article Galaxy works with Curedatis, and then some of the additional tools within Article Galaxy, and more specifically our references product, how those tools work together, and then also how Article Galaxy can supplement some of the functionality that Robert talked about, and also some of your workflow needs when it comes to literature, organizing things, that sort of stuff. So I'll walk you through some of these processes and really how our tools can really help work for you. So on my screen right now, if you're not familiar with Article Galaxy, is a common kind of dashboard, just a standard layout. There's a bunch of different tools and options that can be configured here. Article Galaxy as a product really focuses around helping users discover literature, so doing things like searching for content. And then we also help users acquire literature at the lowest possible cost, so doing things like finding open access articles, helping organizations surface content that you subscribe to, things like this. And then also we do things like help organizations manage that literature. So this is really playing into Article Galaxy References, which is a relatively new product that I'll introduce you to today if you haven't seen it. So let me click on the references link here, and then let me talk a little bit about a few things related to really what Robert was talking about, how Curedatis works and how Curedatis works with Article Galaxy. You saw within the workflow that Robert showed, you go through a process of screening this content. So you're looking at abstracts, and then you get to a point where you say, I want to get those PDFs, whatever they might be. You can order them really with like a one-click seamless access. All of that content is distributed through the Article Galaxy delivery engine, automatically into Curedatis. That just happens. It's a real seamless sort of process. But an important piece to understand is that that content's also flowing into your organization's PDF library. So what we're looking at here in Article Galaxy References is a PDF library for organizations. This is built on a lot of copyright compliance functionality and things like that, which we can always answer questions on. But ultimately, this content flows in here automatically. So what you see on my screen is a bunch of literature that just kind of automatically happens with your users and any of the content that you would order from Curedatis or even directly within Article Galaxy. So Robert was kind of showing an example of knee replacement and some device discussion around that sort of stuff. So I can come into this PDF library and search for literature around knee replacement. Maybe it's a topic that I'm working on in Curedatis. Maybe I want to look at a different topic that's maybe related and then do some work in Curedatis. I can easily search for this content, find all those items that are related to knee replacement, select an item, and I'll get information like the citation details. I can save the reference, check reuse rights, share links to this content. I can also look at things like the abstract. So I'll get a pretty good understanding of what this article is about. Keep in mind, this is your organization's PDF library. So if you want to just open the PDF, review it, you can do that. These Article Galaxy and Curedatis were built with the same functionality when it comes to doing things like annotating, so marking up PDFs, that sort of stuff. So you can open these PDFs and make notes and that sort of thing. So the tools are the same, a lot of similar functionality and things within these products. So I can find that literature, and then from here I can begin organizing that literature. So I can select a single item and I can move it over to a folder, or I can select multiple items on a page, or all if I want, and move them into a folder. So all that stuff flows automatically into Article Galaxy, as I mentioned. As a user, you can start looking for that literature and begin the organization of that content. Once you put things into folders, you'll notice that you have a personal folders, your folders. So there are things only you see, and then there are ways to share folders. So I can share with Robert and Julia or whoever in my company, and then we could collaborate around this literature. But what you might do in Article Galaxy is you might create a folder with content that you see here. And you may say, this is relevant literature that I want to get into a project for Curedatis, for example. You can do things like export this content in a whole bunch of formats. Things like RIS are real common, and you can bring that right into Curedatis. You can also get those PDFs if you want to, for whatever purpose you might have. Keep in mind, if you do create a project in Curedatis and the content lives in Article Galaxy, it'll automatically surface that content up for you. You don't need to pay for it again. You don't have to do any of that. It just happens. So that's part of the integration between the two systems. So once you get things kind of organized into a folder, you can create projects. You can do a bunch of things. You can look at the citation information, review the abstracts, open those PDFs up if you want to. You can also do things like tag literature. So if you wanted to tag an item with a specific keyword, you can do that. So maybe you want to tag it total knee replacement, TKR, you can give that item a tag. I'll show you what this does in a second. You can also do things like edit that citation, create notes for collaboration purposes, and then add additional files. So lots of different functionality in here, really designed to help support research needs and organization of that content, and then also work, as I mentioned, with Curedatis to help you with specific project needs and that sort of stuff. When you tag items, what happens is an automatic folder is created. You'll see in here, and they're called tag folders. So you get an automatic folder with TKR. So if I tag additional items TKR, they automatically flow into these folders. So there's ways to kind of create automatic folders within this tool to help you with your needs around literature management and research and that sort of stuff. So some different options in here for doing different things. And then you can also share folders. So as I mentioned earlier, you can create these folders that you can share with colleagues within your organization. So as long as they are part of your organization and in your account, you can find them within the folder, share it, and then you can work and collaborate around the literature within these folders. So pretty intuitive, pretty functional, and lots of good connections with Curedatis and that sort of thing. So this gives you a little bit of an idea about Article Galaxy references and then some of the collaboration with Curedatis. So users often create these folders. They're doing their work with Curedatis. They kind of complete some of that, and then after those steps, often they'll write an article. So they'll want to write a summary of maybe the research, or maybe they're going to write a new scientific article or something like that, or some sort of publication that they're going to write. And Article Galaxy has a built-in Cite and Write tool. And Robert had mentioned this earlier. So I'll navigate over to Microsoft Word and show you this tool. On my screen right now is an attempt at writing an article. What I did is I grabbed an open access article and I kind of just stole the various sections within that article, but I'll give you an idea about how this works. So keeping with the theme really around total knee replacement, that sort of thing, I'm writing this article. Within Microsoft Word, I can connect to Article Galaxy. So there's a real simple add-on. You just click add, it adds it. It says what's your username and password. You put that info in and you're good to go. What happens is it connects automatically to those folders that I was showing you. So I created these folders, maybe they're shared folders, whatever. I can see those folders directly within Microsoft Word. And I can select a folder, in this case, knee replacement folder. It'll pull back all that citation content, gives me the information like the title, author. There's even some abstract information. If you want to click on that, you can. We'll talk a little bit more here in a minute about the Cite.ai integration and how that might be useful for you as well. But what you do is you write that article and then you can inline cite and write. Many of you are probably familiar with this, but you can just put your cursor wherever you want to cite that reference, select the reference, and it will auto insert your reference. There are thousands of referencing styles. I chose just a real simple one so we could see it easy for the demo, but you can change those styles up here. So if you write in APA, MLA, Chicago, whatever style you can create, you can also create a custom style. Many organizations want to do that for various purposes. Bottom line is you'll go through your literature, you'll write your paper, inline cite. If you want to change things, you can. So if you say, that's the wrong citation, you can auto update that, just select a different citation. Then once you're done, you'll go down to your references section and you'll really insert your bibliography or your references automatically. So this is just one item I added, but if you had 10 or 100, it'll just auto load those items and then you're on your way with citing and writing. So really just makes that process much easier. Literature management to reviewing it with some of the things that Robert showed. And then the next step is, maybe writing some sort of review or some sort of paper or article around that content. The next piece that I will briefly show you is related to Cite.ai integration we have. You'll see these little images and these icons in various places throughout our systems. And what they do is it gives you more information about the article. This particular article is brand new, so it happens to not have any supporting literature or contrasting literature as of yet. That will change with time. But what you can do is you can click on these things and continue that research or that work that you're working on. And maybe you want to find contrasting literature related to, well, in this case, total knee replacement. Were there problems, were there issues with products, with users, people in these studies, the outcomes for that sort of stuff? That contrasting literature can be a really great way to learn more about it. You simply click on this stuff, it'll load up that page. And then within this page, you'll be able to look at things like supporting and contrasting literature for various articles. So it's a great way to kind of continue that process of literature surveillance, literature review, research needs, that sort of stuff. So those are really the key things that I wanted to show you all today related to specifically Article Galaxy and some of the tools and functionality and really how our tools can help you with your needs. And even also how Curedatis and Article Galaxy work together. We'd love your feedback. There's definitely areas we can always improve on. With that said, let me bring up the presentation and I will hand it back to you, Julia.
For sure. Thank you. Thank you so much, Robert and Chris, for showing Curedatis and Article Galaxy and the Word add-on. I actually received a question that I should have asked you just a second before you stopped sharing your screen, but I'll just bring this up here.
I can go back.
So all good. Someone from the audience asked, will you show us how this integrates into SciFinder and how many journals allow for reprints to be retrieved as the lowest cost via Article Galaxy? And as an additional comment to that question is, I do all my searching via SciFinder and would like a simple one-stop place to order up any article and would like to download at the cheapest or free price, possibly with a single billing. So
yeah, that's a fantastic question. There's a lot to unpack there, but I'll answer as well as I can. And then if you want to know more, we can definitely set up a call and give you more information. But integrating with SciFinder is very, very easy. They're a preferred partner of ours. So there's a few different ways, but a real simple, easy way we do is when you search in SciFinder, you look for literature or whatever it might be. We can add a button that says, get this article from Article Galaxy. It'll automatically order and flow that content right into the places that I showed you. So it's real seamless. We also have a browser extension, and that tool, it works very well in SciFinder and lots of other places, but it lets you do your work in SciFinder, click a single button, and then that button will say, here's all that literature that you're looking at in SciFinder. Here's how you can get it. And here's how you can get it at the lowest cost. So it'll say things like, this one's open access. This one has a cost. You already own this one. It'll give you that information right there. All of our systems are designed to get literature to users at the lowest possible cost. And then there was a mention in that question or series of questions about billing and end of the month. Those are all things that we can get you set up with. So yeah, I'd love to talk more on it, but definitely can do some integrations with SciFinder to make your life and processes much more seamless and easy.
amazing. Thank you, Chris. And thanks for that awesome question. If you have any follow-up questions, feel free to add it in the chat or in the Q&A.
Yeah.
So I'm adding some links in the chat here right now, kind of talking about next steps. So if you're interested in learning more, you have some additional questions that we didn't answer, you would like another demo, would like to bring someone from your team on, I've sent a link here in the chat to all of you, researchsolutions.com/sales. Feel free to reach out over that link. That'll put you in touch with one of our experts. Schedule a call, get all of your questions answered. And you can also ask for a free 14-day trial. So you can do that for Curedatis, Article Galaxy, references, or all together. So just let us know what you're interested in, and we're happy to get that set up for you. If you are already a client and you saw something that maybe you don't have unlocked as we speak right now in your account, feel free to reach out either to your account manager, or if you don't know who that is, you're unsure, you can reach out to the email that I sent, which is customersupport@reprintsdesk.com. So you can reach out either ways. We'll make sure to connect you with the right person and get you set up. Yeah, just see another question. Yes, there is a trial period. We have 14-day trial. If that's not enough, we can talk about it as well. So yeah, let's get that trial set up for you and as many people in your company that you like. So then let's dive into the Q&A. I do see a couple of more questions came in. The first one is for you, Robert, I believe. Does Curedatis automatically look up PDFs that I already own within Article Galaxy?
Yeah, so I think that is what also Chris mentioned briefly. So if you have it in the company library, so then you will see the sign when in Curedatis. So that's available via your library, and you just need to click that, and this will fly into your project, and there's no need to rebuy that. Yeah, correct.
Awesome. Yeah, good question. Here's another one. Is it possible to purchase Curedatis or Article Galaxy alone?
Yeah, great question. And I mean, the answer is yes. You can go either way or both. And, sometimes you want to just one, you want, and you want to try the other. You can definitely do that as well.
Awesome. Another one here is, will Curedatis support other recommended annotations than the Picoterms?
Mm-hmm. So right now we have the AI trained on the Picoterms, so the population intervention comparison outcome. So there's a room for improvement then in the future as well for those AI labeled, but we have also the dictionaries, which can be specified by the customer. So you can easily prepare a text file, and this can be then uploaded into Curedatis. And so this will be keyword matching, so not artificial intelligence, but you can highlight your own, your own relevant keywords then in the abstract.
Amazing. Awesome. Thank you. Such good questions here today. It looks like we've covered all. Last chance to add your questions last minute. If you have questions that come up later, as I mentioned, you can use the links that I shared or hit reply to the email that you received about this webinar, and we're also happy to take any other questions offline. So for now, we are wrapped up here. Thank you so much, everyone, for joining us today, joining the webinar. We're really excited and happy that you got to know more about our product family and got to see the products in action. Thank you, Chris and Robert, for showcasing that, and have a good rest of your day or afternoon.
Thanks,
Bye,
everyone. Have a great day.